For our final presentation of the evening, I'm pleased to introduce Dr. Robert Langer. Bob is a David H. Koch Institute professor at MIT. He received his PhD from MIT and did his postdoctoral training at Harvard Medical School. His appointment is in many departments, yet his laboratory is right here upstairs in the Koch Institute. He's one of only 11 institute professors, the highest honor awarded to MIT faculty members, and one of only 7 individuals to have received both the United States Medal of Science and the United States Medal of Technology and Innovation. Many of you are, of course, familiar with Bob and his work. He is, after all, the most cited engineer in history and the founder of more than 24 companies with over 1,000 patents to his name. Bob, you ready?
Thank you.
The computer just froze, so we're going.
Well, it's okay. I'll start without it. That's a. But is the microphone working? Can people hear? So anyhow, what Laura asked me to do is talk about something different, which is how do we take discoveries we make at the Koch Institute or at MIT and bring them to the point where we can commercialize them and get them to patients. And that involves starting companies, and I want to tell you a little bit about that. And then related to 2 projects, the one that you just heard, which I'm involved with Elazar and Laura on, and another one, which I'm involved with Rakesh Jain on, both of which have been funded by the Bridge Project. But to start with, let me just tell you a little bit about my own background and relate how I got into this whole area. So I graduated, and some of you may know this, but I graduated from MIT in 1974 as a chemical engineer. And thank you. And chemical engineers, in 1974, they didn't do cancer research. I mean, they almost all went into oil companies. And I thought that was what I'd do too. But I got actually 20 job offers from oil companies, 4 actually from Exxon alone. But most of the jobs, were like increasing the yield of this chemical or that chemical by 0.01%. I wasn't that excited about it, and I was looking for ways to use my education to help people. And I was very fortunate that, after thinking about it and applying to different kinds of jobs, which, were very, very hard to get for me in teaching and in medical research, that Judah Folkman, who was a surgeon at Boston Children's Hospital, hired me to do postdoctoral work. And I went to Children's Hospital in 1974 as a postdoc. I was actually the only engineer in the entire hospital. And the project that he asked me to work on, for those of you that know him, was angiogenesis. And actually at that time, it was an incredibly controversial idea that blood vessels would be able to go into tumors, and if you could stop that, maybe you could stop cancer. My postdoctoral project was actually to see if we could actually isolate the first angiogenesis inhibitor, which was hard for a number of reasons, not the least of which that people didn't think they even existed. But also that they were often large molecules, we thought, and there weren't bioassays. If you look back at the history of medicine, one of the biggest problems in the history of medicine or biology when you try to find factors is often rate limiting, is having a bioassay. There wasn't any. And we did ultimately develop one. I won't spend that much time on it, but it took 2 to 3 months to do. And to do this, to solve that problem, we had to have a way to have a little what we call slow-release polymers, like little microspheres, that could deliver molecules of any size for months at a time in active form. And it not only had never been done, but when you look in the literature in the 1970s, it basically would tell you that it couldn't be done. And in fact, Dr. Folkman asked many experts in the field about this in the 1970s, and they all said, trying to deliver a large molecule slowly is trying to, for any of you to walk through a wall. Can't do it. The only thing really that I had going for me at that time was I didn't know any of that. So I went ahead and tried to do it, and I developed these little microspheres shown here. Here's one cut in half. We actually published in Nature the fact that you could use this to release molecules of any size for really long times. This enabled us actually to create the first controlled release systems for molecules over 250 molecular weight, which is almost all of the molecules one would want to deliver. It also enabled us to isolate the first angiogenesis inhibitors. That was a paper we published in Science in 1976. By the way, it took another 28 years from that paper before the first angiogenesis inhibitor got approved by FDA. It took billions of dollars and terrific work by people at Genentech and many other companies to make that happen. Today, there have probably been about 20 million patients treated with angiogenesis inhibitors through that work that they did. Anyhow, going back though to our own work in the 70s, when we published this actually, and when I first spoke about it, people didn't believe it either. They said that can't be right, that these molecules are coming out. So we actually did a lot of mechanistic studies when I joined MIT as a faculty in chemical engineering. And if you had a regular polymer like this one, and you didn't put a drug in it, it was like a sheet like this. But what we found is if we put drugs in it and then eluted them out over months and months and months, these structures changed dramatically. And there are these little pores in it. And not only that, we did an awful lot of image analysis. This is just one picture. Turns out that the pores are interconnected. They have tight constrictions between them, and they're incredibly winding and tortuous. So it takes a really long time for the molecules to get through. One way I sometimes explain this to people is it's kind of like driving a car through Boston. In chemical engineering, we have this term called tortuosity, and Boston in this has high tortuosity. And over the years, our students have learned how to manipulate that tortuosity so we could design systems to last anywhere from literally days to many years. Now, Elazar mentioned, Elazar is actually my third graduate student. Is that second? I apologize. Okay, and as he mentioned very clearly, a number of things that I'm going to also point out. One of the things that's really been key, and MIT has been fantastic at this, but to me, that getting patents has been critical to getting what I'll call financial leverage to create companies to ultimately get products made. So Dr. Folkman said to me in 1970s, we should file a patent on this. And we did actually. And 6 years in a row, the patent examiner turned it down. It wasn't because it was wrong or anything. The patent examiner didn't understand science. And the lawyer at Children's Hospital just said, Bob, forget it, it's never going to get issued. But I don't like to give up that quickly. So I kept thinking, how could we convince this guy, the examiner, that he should allow the patent, legally, of course. and I remembered that when we I first started talking about this, everybody told me it was wrong, it was impossible. And I wondered if anybody ever wrote that down. So what I did is you can do what's called a science citation search, meaning you could look back at your Nature paper, see who cited it and what things they said. And actually I found a really terrific quote. I had no idea this was written. I'll just read it to you. This is from 5 of the top scientists in the world at that time. I did this in 1982. It's a 79 quote. It says, generally the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example proteins, could be released by such a technique because of their extremely small permeation rates through polymers, like going through the wall. However, Folkman and myself have reported some surprising, it's a very good word for a patent examiner, results that clearly demonstrate the opposite. So I showed that to our lawyer. He flew down, he was very excited, he flew down to Washington to show it to the examiner. And the examiner said, oh, he said, I had no idea. He said, I tell you what, he said, I will allow this patent if Dr. Langer can get affidavits from each of these 5 people that they really wrote that. And I did, I wrote each of them. They were all kind enough to say they wrote it. And we got this very broad patent. With that patent, we were able to, it's interesting, we actually licensed it to some large companies and they didn't do very much with it. But then ultimately, we got involved in starting companies ourselves, and we also did some joint projects with some companies. And now, actually, there are many products on the market based on this. A number of them are used actually in prostate cancer, advanced prostate cancer. For example, one of the key ones is if somebody had advanced prostate cancer, there are drugs like luteinizing hormone, releasing hormone analog, but you can't swallow that. You can't take it in a patch, it's too big. If you inject it, it's destroyed right away. But now it's put in little microspheres. Originally, they're or rods, and they're originally lasted for a month. Now they're lasted for 4 months. Also, other types of things we did led to new treatments for brain cancer, like the gliadel wafer. These are not cures, but they have led to treatments that have affected millions of patients, and many other diseases too. like schizophrenia, type 2 diabetes. One of the companies actually that I got involved in helping start has been a local company called Alkermes, and we did it with the patents, and with first 4 employees were former students. They have now 25 products that are FDA approved or in clinical trials, and about 1,200 employees. But what we did, and this is a theme that we've done over and over again, is to leverage what we did at MIT or Children's Hospital and bring it to these companies, which then create manufacturing plants, and I'll give you a second example now, and then bring it to the clinic. The second example I want to give you is nanotechnology. This actually started with another paper we wrote in Science, and then in 1994, and then Omid Farrokhzad came to, who's now at the Brigham, joined our lab as a fellow, and we got actually a Koch starter grant, which Tyler was kind enough to give us, which was like $50,000, $100,000 to try to create targeted nanoparticles that you could decorate just the right way. I'm not going to go into the science too much, but that you could decorate just the right way to target to a tumor. I never explained this so well, so but I've actually been lucky that Nova, the TV show, they came here and did some filming of what we did, and they explained it 100 times better than I do, so I'm going to show you their video of us.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That, in turn, gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered. A key that will only fit the lock of cancer cells.
You can imagine I can't duplicate that. And I should say that Tyler and a lot of the clinicians that I work with tell me it doesn't really blow the cell up quite like that. And what we did, it's now been tested in well over 100 patients. It's still early in phase 2 trials, and again, a lot of experimentation needs to be done, but it does appear safe, and these are some early indicators with CAT scans of efficacy. In a paper we published in Science Translational Medicine, where you see tumors here in the lung, and then they go away 42 days later. Here's another example. The company we started out of here is Bind Biotherapeutics, and they've created whole new manufacturing plants to make nanoparticles. Never been done before. It's a major challenge building on work that we did from the lab, and it took a lot of money. This is just a little bit about Bind. We had papers in Science and PNAS. Again, critical to this were broad patents. 3 rounds of financing, plus NIH, NIST grants, over $100 million raised, started clinical trials in 2011. But this is the leverage that basically you get, now once you start to get data and into patients, then really big companies come and say, well, maybe they'll try their drugs in it. And so Amgen, Pfizer, AstraZeneca, there'll be another one soon, all did substantial arrangements with Bind to look at their drugs, and they went public in 2013. Now, I go over this simply to point out that this is a model we've used over and over again. The New York Times actually wrote a big article on it about 1.5 years ago, and it's led to many companies, many medical products, many new treatments for cancer and other diseases, also thousands of jobs. I mean, and it's not just what we do, it's like it's a lot of MIT, but you look at the area around Kendall Square, right around where we are. I remember being a graduate student here, like I said, well, this was a slum around here. It was terrible. But today, it's unbelievable. You look at all the companies, the hotels, the restaurants, they came as well from this whole infrastructure that MIT has done. Now, the Bridge project, I just want to go over it briefly. Both first, what Elazar and Laura have done, and I mentioned that patents are key. So we filed, this is the patent. I'll just expand a little bit on this, but basically the key is always a claim. It's a method for treating a tumor of the pancreas, biliary system, gallbladder, liver, small bowel, or colon, comprising basically just what Elazar said, of deploying a drug-eluting device in a tissue site of a patient in need of treatment, and has a few other things. And it releases a therapeutically effective amount of the chemotherapeutic drug. This is key, comes right out of the work that Elazar just gave in his last talk. And the strategy that Elazar and Laura and the colleagues have done is outlined here. And basically, they figured out some relatively inexpensive ways to do to move it forward using doing large animal studies and then clinical trials. And in fact, Laura was MIT, as many of you may know, has this 100K competition, and Laura actually created a team for that and finished second in the health category. Should have finished first, but she did terrific. And so this is something that we hope will move forward to the clinic. The other area, the Bridge project, is something we had started with Rakesh Jain, and that's to create nano ARBs. These are angiotensin blocking agents, which are already approved, but that we could put maybe in nanoparticles along with different drugs to create improved drug delivery in pancreatic cancer. And this also relates to something Elazar said. Elazar mentioned that that the big problem in terms of trying to get to pancreatic tumor is the stroma. It's hard to get drugs in, particularly large molecule drugs in, because you get collapsed tumor vessels and all this stroma, and you have this collagen matrix. But what you could do is there are drugs, it turns out, and Rakesh has done some wonderful work in this, my collaborator, that there are drugs that are actually already FDA approved, these angiotensin receptor blockers that you could actually use for treating heart disease and other things, and if you give them, totally changes the stroma. And our thinking is that if we could then open the tumor up, so to speak, then you could get the drug in because it's perfused so much better. And so what we did, and this is just really early work, in fact, where you can use different animal models now and show that when you do some of these combination therapies, you can increase survival significantly in different animal tumor models. And again, this is not optimized at all, but it basically shows that you can prolong life in these, and we're working on doing that better. Once again, critical in this is having a patent, as I mentioned. Here, the patent is a method of improving the delivery or efficacy of a therapy in a subject. I apologize for all the legal language, but I'm just trying to be accurate. Comprising you identifying the subject who needs it, and you give it, like I say, an antihypertensive or collagen modifying agent. And like I say, the nice thing about these is they're already FDA approved. And then you either could administer it to the subject, administer with, these are all ORs, or administer this microenvironment modulator, which it also is, improving delivery or efficacy of the therapy in the subject. What we've done, actually, is, again, because we want to, again, the Bridge thing has been a great catalyst that gets us to the point where we've been able to get some interesting data, but what we wanted to do is, again, try to see if we could get it to the point where we can do what we did with Alkermes Bind and those other companies. So what we did is we got a terrific CEO, Alan Crane. He's a partner of Polaris. He actually is CEO of Momenta, which is another company that came out of actually my work with Ram Sessaskaran, who's also in the Koch Institute. It's been a terrific company, and also he's CEO at Cerulean. They're both public companies, and he was a senior VP of business development at Millennium before that. In fact, Alan just presented, Johnson & Johnson had what's called the Quickfire Challenge, where they had 60 new companies compete, and he actually, the top 3 actually get free lab space for a year, so he actually won that, and Polaris has seeded that with $1.5 million to get this started. So again, these are very, very early, but we're excited by both the work that Elazar and Laura have done, as well as the work with Rakesh, that these are even at the point where you can even start thinking about companies, because it's a long, long road. But we hope by doing that will ultimately raise the kind of money, the kind of manufacturing ability, the kind of ability to have extensive clinical trials so you can understand what the right kinds of stents are, the right types of ARBs are, and so forth, so that we can ultimately come up with better treatments. Just like everybody, I really want to thank the Frontier Funding Program from the Koch Institute, the NCI, and the Bridge Project. We would not be able to do that, any of this, without that. And it's really been those key grants, as I've mentioned over the years, that have enabled us to get started whenever we've had new ideas that we could help could change the course of medicine. Thank you so much.